Hey guys, so today we're completing the 2021 A-Level Paper 1. So our first question is about enzymes, asking us to describe the induced fit model of enzyme action and how an enzyme acts as a catalyst. So there's two parts to this question. First, let's describe induced fit. This is basically when an enzyme substrate complex is formed. So obviously the enzyme binds to the active site. The substrate binds to the active site of the enzyme. But then induced fit leads us to suggest that the active site of the enzyme actually changes very, very slightly in order to fully fit the substrate. And then for the second part of our question, which is and how an enzyme acts as a catalyst, we know that as like in any case, all a catalyst does, well, maybe not all a catalyst does, but a catalyst reduces the activation energy needed, making it so that the reaction can occur faster, essentially. Okay, so if we carry on, our next question is basically about ATP synthesis. If you just remember the equation, which is that ADP plus PI reacts to make ATP. So therefore, if they have ATP synthase, our enzyme, and then we have our buffer and our substrates, all we need is ADP, also known as adenosine diphosphate, in order to complete this reaction. Okay, so now we have a graph. This is an enzyme graph, which you probably recognize. As we can see, we have our typical increase, which is followed by our plateau. So the first question is asking us to suggest and explain the procedure the scientists could have used to stop this reaction. Okay, this is calling on our basic enzyme knowledge. So think, what will stop enzymes from working? Obviously the cold, which is the one that I used here because it causes a decrease in kinetic energy. But you could do other things, such as you could boil the solution in order to denature the enzymes, so then enzyme substrate complexes cannot be formed, so then the reaction cannot go ahead.